So you might think that why we are manipulating and how we are manipulating our DNA and how we are also cutting the DNA. Okay, so there is a story. Once there was a bacteria. He was living happily. He fears no one. And someday he meets his rival, that is a bacteriophage. Bacteriophage is a virus that only attacks bacteria. Okay. So he one day meets his rival, bacteriophage, and bacteriophage just sits on the bacteria and introduces its viral gene. Viral gene gets introduced inside the bacteria, and now this gene hijacks the machinery of the cell and thus kills the bacteria. তো ব্যাকটেরিয়াকে মেরে ফেলল। তো এরকম দেখা যাচ্ছে for a long period of time the bacteriophage is constantly killing the bacteria. ওকে, সো তো ব্যাকটেরিয়াও তো মান সম্মান আছে, ওরও খারাপ লাগছে যে আমাকে বারে বারে এসে মেরে দিচ্ছে। So bacteria gradually grows a system. আমাদের বডিতে যেমন ইমিউন সিস্টেম রয়েছে, so the system we are going to discuss is the immune system of that bacteria. That is the restriction modification system. Okay, so in restriction modification system, the bacteria carries two sets of enzymes. One enzyme called restriction endonuclease. One is the restriction endonuclease and another is called modification enzyme. So after long humiliation, the bacteria finally decides to get some armors for itself. That is, first armor is restriction endonuclease and the second armor is modification system. Nobody will ask you how the RMS system works, but still to understand the process we should discuss it. Okay, so in the restriction endonuclease, what it does, endonuclease symbolizes any enzyme that can degrade a nucleic acid. So restriction endonuclease degrades nucleic acid. So if now the bacteriophage tries to incorporate its own viral genome inside a bacteria, the bacteria what will do? The bacteria will produce the restriction endonuclease and which will kill the viral DNA. So then the bacterial cell says apun hi bhagwan hai, kyunki nobody can kill him. Okay, so there is a question arises that the bacteria itself has its own DNA. So if there is restriction endonuclease and it is degrading the nucleic acid, so how the bacteria's own nucleic acid is protected? The question simply arrives, না যে কি করে আমি যখন একটা এমন এনজাইম তৈরি করছি যেটা কিনা আমার নিউক্লিক অ্যাসিডকে মেরে ফেলে, তো হাউ ক্যান আই প্রোটেক্ট মাই ওন নিউক্লিক অ্যাসিড? দ্য অ্যান্সার লাইজ ইন দ্য সেকেন্ড পার্ট, দ্যাট ইজ মডিফিকেশন এনজাইম. সো look, the bacteria is very much clever, na? So what he develops a modification enzyme. So what he does, this modification enzyme modifies the bacterial chromosome. Okay, so if there is this is the bacterial chromosome. So in C5 or something, in case of methionine, that is N4 nitrogen 4, মানে অ্যাডেনিন বেসের 5 নম্বর কার্বনে আর কোনো সময় 4 নম্বর নাইট্রোজেন বেসের সঙ্গে কি করে? মিথাইলেশন occurs. Methylation occurs. So what the methylation will do? Methylation will indicate the restriction endonuclease that এটা আমার chromosome. Okay, so when the restriction endonuclease is going to kill a DNA, as it has no eyes, it cannot differentiate between what is viral DNA, what is self DNA. So when it finds that a DNA is methylated at certain positions, it leaves the DNA. Means it does not kill the DNA. It kills the other DNA that has no modification. So as the bacteria is modifying its own DNA to protect its own DNA from the restriction endonuclease, the system called modification system, and the enzyme that is responsible is called modification enzyme. Okay, so this is how the RNA system developed, and when scientists were searching for an enzyme to cut the DNA, they found this very beautiful enzyme. And what is the beauty of this enzyme? That it only cuts into a specific site. So restriction endonuclease cuts into a specific site that is called restriction site. Restriction site. Okay, so let me draw a bit, erase a bit. So we have our bacterial genome. Okay, we have our viral genome. We have our viral genome. We have modified our own DNA to protect against the monster, that is RE. This is restriction endonuclease. And which site is modified? The same site that is also present in here. Means both the genome contains the restriction sites. Both the genome may contain the restriction site. So if the restriction site is present in its own genome, then it has a probability to cut its own DNA. So it modifies its own DNA and leaves the viral DNA unmodified. So now the monster comes and identifies the unmodified DNA and chops it off, and thus protects the bacteria from viral attacks, bacteriophage attacks. So when scientists found this enzyme, they were very much happy because of two reasons. First reason is it is an endonuclease. I am using a term restriction endonuclease. Endonuclease. So what endonuclease is that? Endonuclease cuts the DNA in the internal sites. Internal site means it can cut here, it can cut here, it can cut here. Okay, no, it will not cut here. It can cut here, okay, but not at the ends. Basically, it finds an endo sequence that has the similarities which it can recognize, which is the restriction site, and then it cleaves the part. মানে আমার একটা ডিএনএ এর মধ্যে যেখানে যেখানে রেস্ট্রিকশন সাইট রয়েছে সেখানে এন্ডোনিউক্লিয়াস কে কাট করতে পারবে। বেসিক্যালি এই রেস্ট্রিকশন সাইট গুলো কোথায় থাকে? ডিএনএ এর ভেতরে থাকে, ঠিক আছে? ওকে, সো ফার্স্ট কজ ইজ it is an endonuclease. Second cause is the specificity. Specificity means different types of restriction endonuclease recognizes different types of restriction sites. Let me give you some example. That is, one restriction endonuclease is called EcoR1. So EcoR1 is a restriction enzyme, and it only cuts the sequence GAATTC. So when EcoRY counters a sequence inside the DNA, that is this sequence, it introduces two cuts. One is here, one is here, and the DNA is cutted into this, like this. So this is how the sequence is cutted. This part gets cut and this part gets cut. Okay. So this is how one restriction endonuclease cuts into a. There are also several sites that is there is another enzyme called PST1 that that cuts into a site called CTGCAG. CTGCAG. It cuts from here. Okay, so these are several restriction enzymes, and these are several restriction sites. These are restriction sites, and these are restriction enzymes. Okay, so you are understanding that the biotechnology that we are discussing in this part has its soul in this enzyme, that is the restriction endonuclease. Without restriction endonuclease, you cannot cut a DNA, and if you cannot cut a DNA, you cannot generate the target DNA or the gene of interest, and you cannot proceed further. Okay, so this is the RMS system, and this is some examples of endonucleases. I will also encourage you to know some other enzymes, that is EcoR1. Please do some research on them. EcoR5, HIND3. Oh, these two are very popular. Also, SMA1. Okay, these are some enzymes, and also you find out what are the restriction sites for these enzymes. Okay, this is your HW. Okay, so we were discussing the enzymes. We are looking into the enzymes. So what enzymes are required? So there are two sets of enzymes. One is the procedural enzymes, that we are going to, those enzymes that is required to follow the procedures, and there is some end modification enzymes. End modification. I am just classifying. There is no such classifications. I am just classifying them according how I am going to represent it. Okay. So the enzymes that is required for the work are simply a polymerase. Because if there is no polymerase, there is no multiplication of the target gene. Usually we are talking about the DNA polymerase 1, DNA pol 1, and also there is one Taq polymerase. Taq polymerase. Taq polymerase is used in PCR, which will be discussed in the next part. And DNA polymerase is the one that helps in the replication of the vector molecule. Okay. There are some nucleases. Nucleases are the enzymes that can cut a DNA or can degrade a nucleic acid. So there are several types of nuclease, can be the Mung-bean, Mung-bean. Okay, one example. Another example is RNase H, RNase A, and there is endonuclease. So let me give you a question, that is, what is the difference between nuclease and restriction endonuclease? If you have enough time, do comment your answers. I will check them. Okay. So endonuclease, and there are certain types of restriction endonuclease we have already discussed in the previous section. And there is ligase for joining the parts. Okay. And in modification enzymes, there are several enzymes also. In modifying enzymes, there is one enzyme called terminal nucleotide transferase. Terminal nucleotide transferase. There is alkaline phosphatase. There is polynucleotide kinase, polynucleotide kinase. It is usually T4 polynucleotide kinase. T4 is the bacteriophage from which it is obtained. Okay, and there are no more names. These are all the end modifying enzymes. So what these do, what these enzymes usually do. So I am drawing it. There is no space. Let me draw here. Okay, I have already told you this part because this is simple. This one is simple. I need polymerase to multiply my vector molecule. I need nuclease to cut the target molecule where it is situated. If it is situated in the end, we will use exonuclease. If it is situated in the middle, we will use restriction endonuclease. Okay, this is basics. And then ligase. So we have our gene of interest, and this is our vector. We need to incorporate this gene of interest and need to seal this and need to seal the portion with the vector molecule. So the sealing process is done by the enzyme ligase. Okay, ligase is also used from T4 ligase. This part is interesting. What is terminal nucleotide transferase? Terminal nucleotide transferase is a template independent polymerase. So this is our, suppose let me say GAATTC. So this is a sequence, and if I want to produce a sticky end, I will use this enzyme, and as it is a template independent polymerase, it will adds up new bases in the tail without even a template. Okay, so this is usually poly A tail or poly GC tail, whatever nucleotide you have given it. It is T. Okay, so this is how the ends are modified. So this is end modification system. What end modification system? The target DNA that we have isolated from a DNA, and we are modifying the ends of the DNA for the sake of to incorporate into a vector easily. আমরা এই যে টার্গেট ডিএনএটাকে আইসোলেট করেছিলাম, এবার অনেক সময় কি হয়, আইসোলেশন তো ভালোভাবে হয়ে যায়, কিন্তু একে একটা ভেক্টর মলিকিউলের মধ্যে ইনসার্ট করতে গেলে ঝামেলা শুরু হয়ে যায়। তখন আমরা কি করি, তখন আমরা লাস্টের দিকে মডিফিকেশন করতে শুরু করি, যে এটা একটু কেটে ছোট করে দিলাম, একটু কেটে বড় করে দিলাম, বা এরকম স্টিকি এন্ড তৈরি করে নিলাম, তখন আমরা এরকম ভাবে ইউজ করে থাকি। ঠিক আছে, সো দিস ইজ এন্ড মডিফিকেশন। This is the terminal nucleotide transferase. Okay, then there is alkaline phosphatase. So phosphatase is any enzyme that removes phosphate from the 5' end. So this is suppose this is a stretch of DNA, this is the 5', and you know that there is a phosphate group in the 5' section, and we are removing it by the help of the enzyme alkaline phosphatase. So what alkaline phosphatase will do, it will chop off the phosphate groups from the 5' end. And what the T4 polynucleotide kinase does, just the reverse of what the alkaline phosphatase does. It adds up the phosphate group. So PNK, it is also referred as PNK. Polynucleotide kinase, what it does, it adds phosphate group at 5' end. Okay, suppose I have a gene of interest, I have two or more gene of interest. So if there is a phosphate group present, and also there is a phosphate group present in both the cases, what can do, if there is a 3' OH, the 5' and the 3' will merge and will cause a nuisance. Okay, মানে আমার কাছে দুটো সিকোয়েন্স ছিল, এবার প্রথমটার 3' OH এর সঙ্গে পরেরটার 5' OH যুক্ত জোড়া লেগে গিয়ে একটা ঝামেলা করে দিতে পারে। তাই জন্য আমরা অনেক সময় যখন দুটো বা তার থেকে বেশি টার্গেট ডিএনএ ইউজ করি, তখন আমরা কি করি, তাদের ফসফেট গ্রুপগুলোকে সরিয়ে দিই। ঠিক আছে, ওকে, দিস ইজ দ্য enzymes have covered, RNA system has covered. there is one topic which is called star activity. So star activity is all the enzymes that I have mentioned here requires a optimum physical condition, optimum condition. It can be pH, it can be temperature, whatever it is. Star activity refers to the activity of these enzymes when there is no optimum condition is provided. The enzymes, usually the restriction enzymes, sorry, I have mistaken previously, not all the enzymes, only the restriction enzymes. Let me erase it. Eco R1, and it cuts the sequence GAATC. Okay, so this is the sequence which this restriction enzyme recognizes, and if there is not optimum conditions are present, suppose the temperature is way more high or the pH is very much low, so then the eco R1 will not cut the exact sequence, but rather it will cut a similar looking sequence. Suppose the sequence which it will recognize GAATC. Suppose this sequence. So originally it was recognizing this sequence, but under altered conditions, the eco R1 recognizes this sequence. So this altered activity is called star activity. Okay, it helps the scientist, or it helps the students to remember that the one enzyme only functions when there is proper physical environment is provided, that is an optimum temperature, an optimum pH, and optimum buffers are all required for the proper function of the eco R1 enzyme. Okay, so this is the enzyme part, and now let us move on to the next part, that is nomenclature. Now we'll go to the next part, that is the nomenclature of the restriction endonuclease. Restriction endonuclease এর নাম কিভাবে দেওয়া হয়, না আমি আগেই কটা এনজাইমের নাম বলেছি, যেমন eco R1, and the process by which they are named is the abbreviations of the genus and the species. Means this enzyme comes from the bacteria called E. coli, Escherichia coli. So the first letter of the E and the second two letter of the species, first letter of the genus and second two letter of the species. So eco, R is the strain, that is RY13. RY13 is the strain which is considered here, and this Roman number indicates the order of the same bacteria, same enzymes which are discovered later, just like eco R5. So this is the first enzyme, this is the fifth enzyme. So this is the order which is mentioned here. Okay, so this is how the nomenclature works, and it is important from the exam points of view. Okay.